Welcome back to Peking University MOOC Bioinformatics Introduction and Methods. Let's continue with our Week 9 lectures. In the previous units, we learned about the concepts of ontology, the hierarchical structure of the gene ontology and KEGG pathway database, how they are associated with genes and gene products, and how the Cobas software annotates and identifies statistically significant pathways. In this unit, I will use an example from my own lab to illustrate how we can apply these ideas and methods to study a real problem and discover interesting patterns. As many of you know, addiction is a serious medical problem. There are many substances that can cause addiction, such as cocaine, heroin, tobacco, alcohol, and so on. As this figure on the top right shows, different addictive substances vary in terms of the level of physical harm and dependence they cause. However, despite the differences, addictions to different substances seem to share a common course. So a question that we and other researchers had wondered about was, is there a common molecular pathway underlying addiction? We felt that to answer this question, a bioinformatics approach might have a unique advantage. Most of this work was done by a talented former graduate student of mine, Chuanyun Li, who is now a faculty member at the Institute of Molecular Medicine at Peking University. To answer this question, we first gathered genes that are related to addiction. Chuanyun read about 1,000 papers and collected all the genes that had been linked to addiction in literature. There are two main types of experimental evidence. The first is evidence from genetic studies such as linkage analysis and association studies, and the second is evidence from molecular biology experiments such as gene expression and proteomic studies. These two types of evidence each identified hundreds of genes related to addiction. However, only 4 genes are identified by both types of experimental approaches. The overlap is surprisingly small. This kind of discrepancy between genetic and molecular biology findings had been previously reported by other groups about other complex diseases as well. After the initial confusion, we decided to look into this problem from other different angles. Eventually, we found out that even though genetic and molecular biology approaches identified very different genes, the genes tend to fall into the same pathways and connected protein-protein interaction networks. They just tend to fall into different parts of the pathways and interaction networks. Based on this observation, we decided that in order to get a comprehensive picture of addiction, we need to integrate gene sets from different types of experimental approaches. This resulted in a set of 1,500 genes. Because there may be noise in this large gene set, we selected a subset of 396 genes that were supported by 2 or more evidences. Now, given the set of 396 addiction genes, which pathways are involved? Which pathways are statistically significantly represented? You may remember that Cobas is one of the tools for this type of questions. So we ran Cobas on the 396 addiction genes against a background of all human genes, and found that 18 pathways were statistically significantly enriched, including long-term potentiation, long-term depression, gap junction, neuroactive ligand-receptor interaction, and so on. But we haven't yet answered the question, are there common pathways underlying addiction to different substances? Please pause a moment to think about how you might do this. The way we did it was this. When we collected genes related to addiction, we collected not only the data, but also the metadata, meaning that for each gene linked to addiction, we also collected the experimental details, such as the type of experiments, the parameters, the species, the brain regions when available, and so on. In particular, for each gene, we collected metadata on the addictive substance studied in the related experiments. Once we had put all these data and metadata into our relational database, we could easily write Perl, MySQL, and R programs to analyze the data. We focused our analysis on 4 substances that had the most data: cocaine, alcohol, opiates, and nicotine. After we ran Cobas 4 times on the 4 substances one by one, we identified these 5 common pathways that are statistically significantly enriched for all 4 substances. Interestingly, 2 of the pathways, the GnRH signaling pathway and the abduction pathway, had never been linked to addiction before. But how could we have discovered 2 new pathways related to addiction without doing any experiments? The reason is that if you look at the genes in these 2 pathways closely, you would find that each of the related genes had been found in some lab somewhere in the world using some experimental approaches. However, because each lab saw only one gene, they could not draw any conclusions about the pathway level. Only after you apply the kind of bioinformatics analysis that we talked about this week on the kind of data and metadata represented in the computer with the hierarchical structure that we talked about this week, could you suddenly see these interesting patterns. So this is a good example of the power of bioinformatics in making biological discoveries. In summary, to facilitate communication and computation, we need to store data in database whenever possible, define an ontology for the data, and collect metadata together with data. To discover higher-level patterns in a set of genes or gene products, we can identify the most significant pathways and functional categories by performing statistical analysis with tools such as Cobas. I hope these lessons can be useful in your own future research. Thank you for your attention.